Hello, my name is Margit Tolle, and the title of my today's lecture is the effect of silicon on the organically grown cucumber transplants growth and quality. Short introduction. Silicon is the second most abundant element after oxygen in soil. Silicon dioxide comprises 50 to 70% of the soil mass. All plants rooting in soil contain some silicon in their cells and tissues. Supplying crops with adequate plant-available silicon can suppress plant disease, reduce insect attack, improve environmental stress tolerance, and increase crop productivity. Silicon plays important roles in mitigating the biotic and abiotic stresses. Biotic stresses are insect pests and pathogens. Abiotic stresses are metal, salinity, drought, chilling, freezing. Purposes. The purpose of the investigations was to look the effect of silicon on the cucumber transplant quality because only few investigations have been carried out on cucumber transplant treated with silicon. Another aim was to see whether there is the effect of silicon so early in plant development. Materials and methods. Experimental site was Estonian Crop Research Institute. The variety Landora F1 was used and crop was cucumber. There were two treatments, stabilized silicic acid treatment and control. Sowing on 21st of March 2014 in plastic trays and seedlings were grown in heated glass greenhouse. Seedlings were transplanted once into individual pot on 10th of April 2014. Seedlings and transplants were grown in Novarbo B2 organic biolan substrate for organic cultivation. Each treatment consisted of 16 plants. Within that, one plot consisted of 4 plants in 4 replications. The experiment was repeated at the same time. The results were gathered on 12th of May 2014. The greenhouse lighting at plant level was approximately 12,000 lux from high-pressure sodium lamps. The plants were additionally lighted in the period of 18 hours. The plants were grown with a minimum day and night temperature of 20 and 18 degrees respectively. The height of cucumber transplants in silicic acid treatment was higher than in control plants. The stem diameter of cucumber transplants in silicic acid treatment was higher than in control. The content of nitrates in raw cucumber transplants was higher in silicic acid treatment than in control treatment. The content of nitrogen in cucumber transplants dry matter was higher in silicic acid treatment than in control treatment. The content of phosphorus in cucumber transplant dry matter was higher in silicic acid treatment than in control treatment. The content of potassium was not significantly different. The content of calcium in cucumber transplants dry matter was not statistically different. The content of magnesium in cucumber transplant dry matter was higher in silicic acid treatment than in control treatment. Here you can see the cucumber transplant in the experiment. On the left-hand side, silicon treated plant, and on the right-hand side, control plant. Conclusions. Cucumber transplants were taller and stem diameter was greater in silicic acid treatment compared to control. The content of nitrates, nitrogen, phosphorus, and magnesium were higher in silicic acid treated plants. Acknowledgement. The present research was carried through with financial support from Estonian Agricultural Registers and Information Board with the help of the Jagma Agro OÜ and Estonian Crop Research Institute. Rexil Agro BV have supported by free of charge silicic acid, provided the training for using SAT technology and issued some travel grants. You can further on read about it in my book about silicon as a challenge for sustainable vegetable crop production, and you can buy it from www.veggiescultivation.com/books website. Thank you very much for your attention.